Hello everyone, I'm Molly Campbell, science writer and editor for Technology Networks, and I'm here to introduce the next talk. New 4D proteomics methods for clinical research, PASEF, dia-PASEF and prm-PASEF. I'm pleased to have Dr. Gary Kruppa joining us as your presenter. Dr. Gary Kruppa currently serves as a Vice President for Proteomics at Bruker Daltonics Incorporated and Managing Director of Bruker SRO. He manages a growing global team of over a dozen applications, field applications and application development scientists, primarily focused on developing new proteomics applications for the timsTOF Pro and supporting customers and collaborators. Dr. Kruppa earned his bachelor's degree in chemistry at the University of Delaware and his PhD in chemical physics at Caltech. Dr. Kruppa has been working with Bruker since 1991, where he first joined as an applications programmer for the FTMS product line, later serving as vice president for FTMS. From 2001 to 2004, Dr. Kruppa ran his own research lab at Sandia National Labs, where he and his team developed novel methods and reagents for chemical cross-linking of proteins for the study of protein structure. He then returned to Bruker, where he served as vice president for business development until 2014. From 2014 until 2016, Dr. Kruppa worked with Professor Christoph Borchers as CEO of prm-Proteomics Incorporated, helping to commercialize its quantitative MRM proteomics analysis of potential biomarkers in plasma, urine, and other biofluids. Dr. Kruppa returned to Bruker in 2016 to serve as Vice President for Proteomics, where he helped launch the revolutionary timsTOF Pro QTOF mass spectrometer, powered by the PASEF method. A warm welcome to you, Dr. Kruppa. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A tab. Please be aware that you can ask questions at any time during the presentation, and we will cover as many as possible at the end of the talk. This Q&A session is your opportunity to tap into Dr. Kruppa's expertise, so I very much encourage you to ask questions. If you experience any technical issues during the presentation, please let us know using the chat button. I will now hand over to Dr. Kruppa.
Okay, I'd like to thank Molly for her kind invitation to speak at this Technology Network Symposium on Translational Omics. My name is Gary Kruppa, and as you know, as she introduced me, I'm the Vice President for Proteomics at Bruker Daltonics. I'd like to start with a brief outline of what I'm going to talk about. The title of my talk is on 4D Proteomics, and I'm going to tell you what that means. And to tell you what that means, I'm going to have to describe the PASEF technique, which is parallel accumulation serial fragmentation, and how it's based on trapped ion mobility spectrometry and how that results in 4D proteomics. And then we'll go on and talk about dia-PASEF and the prm-PASEF methods as well, and then some clinical research applications of 4D proteomics. So the timsTOF Pro and 4D proteomics is a relatively recently begun journey, but we've made remarkable progress, I would say, and there's been a rapid uptake of the technology in the field. The first publication on trapped ion mobility separations was published in 2011, so that's been around for a while. But it was only in 2015 that Matthias Mann and co-workers at the Max Planck Institute in Martinsried, together with people from Bruker, proposed the idea of parallel accumulation serial fragmentation based on trapped ion mobility spectrometry. And I'll describe that and how it really leads to a revolutionary improvement in DDA proteomics on the following slides. So we introduced that at HUPO in 2017, and generated a lot of excitement, I would say, there. And then in 2018 at HUPO, we announced a collaboration with Evosep to take advantage of the speed of the timsTOF Pro for fast proteomics. And we also introduced our first results on DIA acquisition using PASEF, a technique we now call dia-PASEF, which we continued to develop during 2019, showing results at ASMS and HUPO. And at ASMS in 2020, we released dia-PASEF, complete with a graphical user interface and support from a number of software packages to acquire and process the data. We released that at ASMS in 2020, and we're also releasing, as a work in progress, prm-PASEF. So we have a complete suite of proteomics acquisition methods now in PASEF for DDA, dia-PASEF for DIA and prm-PASEF for targeted work. And I will talk about all those briefly in this talk. So first, we, of course, this is all based on PASEF, so we have to talk about what PASEF is. And PASEF is a way to get nearly 100% ion utilization. And the way that that works is shown on this slide. So moving from left to right here, we have ions exiting the desolvation capillary, and they flow through the TIMS separation devices. And then either they can be flow through the quadrupole, and we can measure MS1 spectra, or we can select ions with the quadrupole and fragment them in the collision cell and measure MS2 spectra. Now, how does the TIMS separation work, and where do we get these speed advantages? That's shown in the lower part of the figure here. The two TIMS sections act as ion mobility separation devices. But as opposed to traditional ion mobility, where you push ions through a static gas, what happens here is you have a lot of gas exiting the desolvation capillary together with the desolvated ions. We push the ions to the right, so only the ions and some of the gas enter this tube. All the junk stays in the source region. And because there's millibar pressure here and 10 to the minus 4 on the right, there's a lot of gas flow from left to right. And so the ions are being pushed by the gas from left to right. Now, the TIMS tunnel consists of a series of plates with the same diameter hole, so they form a constant diameter tunnel. We put RF on the tunnel to form an RF ion guide, but we can put a DC potential gradient across these plates. And we do that in a way that we form a potential hill with the electric field increasingly opposing the motion of the ions from left to right. So you have the gas pushing the ions from left to right, and the electric field pushing them from right to left. When those forces balance, they stop, and the position at which they stop is determined by their collision cross-section. The biggest ions feel the most push from the gas, they get pushed furthest up the hill, and the smallest ones stay towards the beginning. So then we drop this potential down, and the voltage at which they exit, we can use to calculate their collision cross-section. More typically, we use a calibration equation to determine the collision cross-section, which is a fundamental property of the molecule. So as they exit, we know what their collision cross-section is. They flow through to the QTOF analyzer. We measure their mass, and then we have their mass, their retention time, their collision cross-section, and their intensity. So those are the 4 dimensions in 4-dimensional proteomics now. So we scan these down and measure all their masses, and in the meantime, we're accumulating the next batch of ions over here. That's why we have 2 of these. This region is just used as an accumulation region. As we ramp this down over the course of 100 milliseconds, we're accumulating the next batch of ions here. When this ramp is finished, we transfer the ions over, and now we do, so that's the parallel accumulation part. We're accumulating in parallel to the TIMS scan. And when we transfer the next batch, based on the MS measurements we did in the previous scan, we can select these ions for MSMS in a DDA approach, and we can select up to 10 to 15 ions per 100 millisecond ramp. So we're doing MSMS at 120 hertz. We do, for each MS ramp, we do 10 MSMS ramps. That total cycle time is typically around 1.1 second, and we typically target 120 to 150 precursors in that 1.1 second. So we've increased our sequencing speed from 20 to 30 hertz with a QTOF up to 120 hertz. But we've also seen a tremendous boost in sensitivity, and that comes from the additional chromatographic focusing effect shown here on the left. Ions that would typically elute over the course of 100 milliseconds now elute in tight 5 millisecond packets, giving big bursts of signal. So we go faster with improved sensitivity, and we keep our 50,000 mass resolution, all the inherent properties of a QTOF. So I like to think of this as a revolution in mass spectrometry. Normally, if you want to go faster in mass spec, you have to sacrifice something. You have to sacrifice resolution or sensitivity. Here, the sensitivity actually gets better. We go faster, and we maintain our resolution, and we get this additional dimension. So it really is a tremendous step forward in mass spectrometry. And this has continued to be a collaborative development between the lab of Matthias Mann and Bruker. The first paper was published in Molecular and Cellular Proteomics in 2018, from which this figure was taken. So where are we now, 2 years after the introduction at HUPO in 2019, approaching 2017, actually approaching now 3 years after release? Well, we can do 6,000 protein groups and 60,000 peptides in a single 100-minute gradient now, with excellent data completeness and DDA. You can see here are 3 replicate runs of 400 nanograms of an in-house prepare a digest of HeLa cells, inject them on column, almost exactly the same number of proteins identified in all 3 runs. And the data completeness, even at the peptide level, is remarkable. 54,000 peptides of the 60,000 for 87% data completeness at the peptide level in this very complex sample. And I'm going to come back to emphasizing that this is a result of the speed of the technology, and speed is important for measuring lots of peptides, not just lots of proteins. We all like to talk about proteins, but peptides are important too. You can use the speed also to go faster. So here we've shortened with our nano, we've been able to achieve a 21-minute gradient in a 28-minute method. So we can do 50 LCMS runs per day with this method. And we can still identify 4,000 proteins and 26,000 peptides in this very short gradient. These would have been considered outstanding results just 4 or 5 years ago with a 90-minute or even an hour and a half, 2-hour gradient. But now we get them with a 21-minute gradient with this technology. So again, you can really do high throughput cohorts, and this is important, as I'll point out later, for clinical translational proteomics. Another thing that's important for translational proteomics is robustness and throughput. You want to be able to run hundreds or thousands of clinical samples and compare the results from sample 1 to sample 999, and the timsTOF Pro allows you to do that. This is a plot of the number of proteins identified from a 200 nanogram injection of HeLa digest, which is used as a QC in the Mann lab. And basically, anytime it drops below 5,000 proteins, you know there's a problem. And that happened, this is in the course of 50 weeks, close to a year. It drops below 5,000 proteins only 3 times, and on each of those times, they were able to get the performance back with the column change. There were some occasional cleanings of the front-end capillary, but in 3 years of operation, we've never had to clean anything beyond the front end or maintain the LC, never had to clean deep into the vacuum system with this system. So it's very robust. We think that comes from that off-axis ion delivery I talked about. The ions are deflected onto the ion beam axis from a perpendicular delivery from the desolvation capillary. Also, the higher sensitivity means that instead of loading a couple of micrograms of sample, we're loading 200 nanograms or 400 nanograms as a maximum. So that helps keep things cleaner. We can tune and calibrate in minutes. And this allows you to run these large cohorts, running hundreds of samples per day, getting thousands of samples done in weeks, and being able to compare sample to sample very effectively. So that's the basic introduction to where we are with PASEF. Now let's talk about dia-PASEF. So it's a unique implementation of data-independent analysis based on the PASEF technology. And it has some really great features, namely that it achieves a nearly 100% duty cycle as does PASEF, but you can also get very high efficiency in fragmenting the precursors, up to 100%. And I'll describe that on the following slide. You also can, because we fragment not just mass windows, but mass mobility windows, we can use the mobility separation to focus on the higher charge states and separate out the +1s, which we're generally not interested in. And that fourth dimension, the ion mobility, we can use for feature alignment. So that increases our confidence in extracting peptides from the library according to 4D feature alignment. This development was actually a result of a conversation between Matthias Mann and Rudi Ebersol, where they realized that you could use the unique features of PASEF to do a very effective DIA experiment. And as I said, we now call that dia-PASEF. So Matthias Mann and Rudi Ebersol. Ben Collins was the main person working on this at the ETH, and he's now a professor at Queen's University in Belfast. And Hannes Rost did a lot of the initial software analysis to be able to process this data using Moby Dick based on the open swath software that he wrote. And these are the collaborators at Bruker who worked on the project. So I just want to remind you what DIA is. In DIA, instead of fragmenting selected precursors, you fragment everything in a mass window. So in this example, we're going from MORZ 500 to 900, and we're isolating 20 mass unit wide windows and fragmenting everything in that window. And in this way, we fragment everything that's there between 500, 100, and 900 in an attempt to get good data completeness, get the same kind of fragmentation, and avoid the stochastic nature of selecting precursors using DDA. But while you're fragmenting, say, the ions from 500 to 520 here, you're throwing away the rest. and the same for every mass window here. So at any given time, you're using only 5% of the ions for fragmentation. And 20 mass units is actually pretty wide. In a complex sample, you might want to use narrower windows to have less complex mixtures of fragments to analyze. And so DIA schemes generally use only 1 to 6% of the ions for MSMS. In dia-PASEF, we take advantage of the fact that there's a correlation between ion mobility and mass. So as we start eluting the ions, the larger ones come out first, and they come out at higher m over z. There's not an exact correlation there, but it's a correlation that we can take advantage of. We set a mass mobility window here, and we fragment everything that comes out first. Then we slide the quadrupole down and the elution voltage down, and we get another batch of ions that we fragment, and so on. And so we can, while we're fragmenting these ions are waiting to elute. So it's a very effective way to use the ions here. We're not throwing away all these ions while we're fragmenting this batch. We pick these off next as we come out, then we pick these off next as they come out. And so that's the idea behind dia-PASEF. And we implemented that. We found that we could, in complex mixtures, we use 4 narrow mass mobility windows. So this diagram really shows you the advantage here. We're fragmenting everything in this window, which is +2 and +3, and we're ignoring the +1 and chemical noise, higher charge states. And if you had only a mass window, this is what you'd select, everything, the chemical noise, the +1. But with the mass mobility, and we should call this a box really, we are selecting just what we're interested in, and we're able to do 4 windows in a scan and make more effective use of the ions. So we can get greater than 4 times better ion utilization typically than DIA, up to 100%. It's selective to ions of interest, the +2s and +3s, and we can get really good data completeness. And as I'll show you, it's qualitative and quantitative. We get IDs and quantitation. Here's just some results from HeLa. In a 90-minute run, we're getting close to 7,700 proteins and 97,000 peptides. This library had 105,000 peptides in it. So at 90 minutes with the speed of the timsTOF Pro, we're getting almost complete library coverage. And even at 30 minutes now, remember with a 30-minute method, with DDA, we were doing around 4,100 proteins. With DIA, we're up around 6,400 proteins and nearly 60,000 peptides. So with DIA and dia-PASEF rather, and short gradients, you can really get very impressive coverage now. And with HeLa, the story is even more interesting because with 90 minutes and 60 minutes, we're covering virtually the entire detectable yeast proteome. So there's not a big difference between 90 and 60 minutes. And at 30 minutes, we still cover, in terms of proteins, a large portion of the proteome and very good peptide numbers. And we have an application note on this, application note 167, where we also show quantitation. I don't want to go into this too deeply, but we mixed E. coli, HeLa, and yeast. E. coli was mixed in at 1/4 of the human. Human was the 1 reference, and yeast was a factor of 2 higher. And you see on the log plot, the quantitation, the peptides quantify along from the different species, quantify well along the expected ratios. We can go even shorter, and we've been working with Evosep on this, and they refer to their methods in terms of samples per day. So at 60 samples per day, that's a 24-minute method. At 300 samples per day, that's a 3-minute gradient, about a 5-minute method. And so you can see that even with a 21-minute gradient, 60 samples per day, we get 5,000 proteins and 40,000 peptides. So you can really start to go fast here and even get reasonable numbers of proteins and run 300 samples per day. There was a poster on this at ASMS, and there's an application note available from Bruker jointly with Evosep on this data. I want to dig into that data a little bit more. This is the 21-minute gradient, 60 sample per day method on the Evosep. On the timsTOF Pro, we loaded 400 nanograms of HeLa. They loaded in this paper where an instrument using FAMES with a compensation voltage of 45, they injected 500 nanograms of HeLa. They showed that the FAMES enabled them to identify more proteins, and they got good numbers. We're a little better, a few hundred proteins more, not spectacularly better. But what you'll notice is that when you look at the CVs, The number of proteins with a coefficient of variation less than 20% is 3,600. We're much higher here at 4,335. So the coefficient of variation on our data, and that carries on to when you look at the numbers of proteins with coefficient of variation less than 10%. So our overall coefficient of variation is much better than they're getting in this paper. And that's, of course, important for clinical proteomics. And I would say that that is due to the speed of the system. We're hitting the same peptides, we're extracting the same peptides more often, and we're measuring more peptides from the same protein, and that's going to give you better CVs. And that's shown on this slide, where at the peak, we're extracting over 4,000 proteins, over 4,000 peptides per minute from the library. At their peak, they're extracting about 2,600 peptides per minute. And overall, we extract, as I said, okay, and as you can see from this slide, we identify 40,000 peptides out of a library that's about 60,000 peptides, as opposed to in the paper I referenced where they identified 29,000 peptides, and they actually had a larger library with the same 60 sample per day method in both cases. And so I think this data really shows that the speed of the instrument is important. It's important in LFQ because more peptide measurements results in better CVs. If you're interested in PTMs, more peptides equals more PTMs analyzed. And for peptidomics, of course, you're interested in the peptides themselves. Briefly want to talk about prm-PASEF because that completes the basically the trio of current shotgun proteomics techniques. We have DDA using PASEF, DIA using dia-PASEF, and we now have prm-PASEF. You can set up target lists, you can target your peptides, but now you target them, instead of using blue mass windows, we now target them using mass mobility windows, and we can do that really fast. So we can target hundreds of targets in an assay in, say, a 30-minute gradient now, because we can do 10 targets in 100 milliseconds. And you can see here's each box represents a compound that's being targeted for PRM in the course of a 30-minute gradient here, 1,800 seconds. And you can see the number of targets per second being targeted. The peak is at around 10 targets per second here. So we think we'll be able to go really fast with really good selectivity and really good sensitivity in PRM. You can see how turning TIMS on really cleans things up. With TIMS off, this is what extraction looks like of 3 different fragment ions. Here's what it looks like with TIMS on, 3 different fragment ions. Then you can see how turning the TIMS on really pulls some of these peaks out of the noise and reduces interferences. And these are peptides, aquapeptides spiked into a human cell digest background at the atomole level shown here on the left. Here's another example showing the effect of TIMS filtering. So you have a really big interference here with TIMS off, turn TIMS on, and this is showing, in this case, not 3 different transitions, but one transition, the Y13, and the isotope peaks of the Y13. And you see turning that on really leads to the expected isotopic distribution for that fragment. And I want to emphasize that even at these speeds, we have resolving power of 40,000. With other instruments, when you go that fast, you're going to be down around 10,000. So that's really going to increase our resolving power. And these results are highlighted on a poster at ASMS, and we'll be coming out with an application note on that soon. And this just shows some of the aquapeptides spiked in at the 5 attomole, the 50 femtomole range. We can get a linear dynamic range of 3 to 5 orders of magnitude, depending on the peptide and the background, but very good dynamic range, very good quantitative capabilities, even at very low levels of spiking. So I just want to wrap up by showing some examples of how our customers are applying this. Roman Fisher, who's the head of and principal investigator at the Target Discovery Institute at the Newfield Department of Medicine in Oxford, recently did a study where he ran 2,600 samples in a clinical cohort characterizing sepsis survival rates in non-depleted plasma. And he also generated a library for targeted extraction. So he ran 1,700 library and QC runs. The stack of plates you need to run all of those samples is shown here. 3 columns were used with the Evosep as the high throughput nano LCMS. they had to clean the ion transfer tube once, so the front end capillary once over the study, but they were able to run continuously with no other cleaning or maintenance over the course of these 4,300 samples. And the data analysis for this is in process, and a publication will be in press. And Catherine Wong recently at the Beijing Medical Institute ran a large cohort study using dia-PASEF for COVID-19. That paper is in press, and she said dia-PASEF has made such a difference that it's become the de facto standard running proteomics analyses, and the robustness is exactly what we need for large cohort studies. So it allowed us to identify and quantify changes not only in individual proteins, but also to protein pathways as a result of COVID-19 infection. So we're very much looking forward to that publication, and thank Catherine for allowing us to use that quote here in this talk. And I won't spend much time on this, but there's another landmark paper showing that collision cross-sections can be learned and predicted by machine learning. This is a paper by Professor Matthias Mann and his collaborators, and he shows that you can predict collision cross-sections and work to include the use of collision cross-sections in library searches to increase the reliability of false discovery rates. Is in progress as well, so we're really excited about this. I just want to point out that since the launch, a wide variety of software packages have taken up being able to handle this data. Skyline supports generating methods for PRM based on target lists and input of DDA data, including collision cross sections, and then data can be re-exported to Skyline for analysis of PRM, and you can also do targeted analysis of dia-PASEF in Skyline. Peaks is our most supported software package. We work very closely with them, and they support very well now DDA, dia-PASEF, and labeled workflows such as TMT. MaxQuant has been improving rapidly. It's very fast now for DDA analysis if you prefer a freely available solution. dia-PASEF is coming soon. Some of the data I showed for dia-PASEF in this talk was analyzed by Spectronaut. They have very high performance and easy workflows for dia-PASEF and library generation. We also work with MS Fragger, Protein Metrics, and Matrix Science. So there's a lot of choices now for processing data from the timsTOF Pro. So I hope I've shown you that 4D-Proteomics with timsTOF Pro is going to be very interesting for clinical research using the three methods I described, PASEF, dia-PASEF, and prm-PASEF, and that we have great sensitivity, reproducibility, quantification, everything you need for clinical proteomics research. And with that, I'd like to answer any questions.
Thank you very much, Dr. Kruppa. That was a fantastic talk. So I know that we are going to have quite a lot of questions and we don't have much time. So we'll move straight on to the Q&A session now so that we can cover as many as possible. So the first question that we have had come through is, can you summarize the benefits of 4D proteomics workflows on the timsTOF Pro for clinical research?
Yes, so as I said in the talk, the speed, combination of increased speed while improving sensitivity are the big advantages. It depends on the kind of clinical proteomics you're talking about. If you're talking about plasma proteomics, sensitivity is not an issue because you have lots of protein, and the speed is the real important thing here. You can get lots of plasma samples from patients, and you want to dig as deeply as you can, as quickly as you can. And we're developing 21-minute methods, 20-minute gradient methods that can dig fairly deep into the plasma proteome. And as the work from Roman Fisher showed, you can process hundreds or thousands of samples in reasonable amounts of time now. If you're talking about proteomics of cancerous tissues, then you're talking about situations where you may have very small amounts of tissue biopsies. And there, I'll add in that the sensitivity is a tremendous benefit. You can start from very small amounts of material. I didn't talk much about it in this talk, but you can even get now reasonable numbers of proteins from a single cell. And if you have a tissue slice with just a few cells, you can still get very good results. So across a broad range of clinical proteomics problems, I think this technology has a lot of advantages.
Thank you for those insights. And the next question that we have is, How can trapped ion mobility and CCS values be used in proteomics research?
Well, to date in proteomics research, we've used the trapped ion mobility mainly as an additional separation and chromatographic focusing device that allows us to go faster and have higher speed. I didn't talk about it much in this talk either, but it also allows you to separate important isomeric species. Catherine Wang, among others, has shown that you can separate peptides that have the same glycosylation on the same peptide, but at different positions by collision cross-section. We've also been able to separate phosphopeptides that are phosphorylated in different positions, have the same mass, of course, because they have the same sequence and the same modification, but the modifications at different sites and they have different collision cross-sections. So the TIMS allows you to separate some things you haven't been able to separate before. And then the last application, I think, is going to be coming very soon, and it results from the ability to generate large libraries of peptides with their collision cross sections, as well as being able to predict them if they're not in a library, and use them in search engines that take advantage of the collision cross section to improve calculation of false discovery rates and give you more confidence in your peptide IDs.
Thank you, Dr. Kruppa. So I think we have time for just one more question in this Q&A session. And so the high throughput dia-PASEF data looks really impressive. You claim 4 times better ion utilization. Can you please explain that better?
Well, it's kind of hard to explain without the slide in front of me, but if you remember the slide, in a quadrupole device, in a QTOF or an Orbitrap, which just says a quadrupole and a mass analyzer, when you're selecting a set of ions or a window of ions for MSMS, you're throwing away all the rest. They're being discarded through the quadrupole. In the dia-PASEF technique, what we're doing is storing the ions in the TIMS tunnel, and then as we start eluting them, we're able to set the quadrupole to pick a window at high mass, while the lower mass, lower collision cross-section ions are still in the trap. And so we're not throwing away ions, all the ions that we're not using. They're still stored in the trap, and then we can scan the trap down, release them, and pick off the next batch. So you can think of it as, I don't know, almost skeet shooting. You know, you pick off the windows of ions as they come out. And if you want to, we have application notes on this. There's a paper by the Mann group on dia-PASEF on bioRxiv that you should be able to find fairly easily. We hope that will be out in press soon. So there's lots of places to read and try to learn more about dia-PASEF. I admit it's not a straightforward concept to explain in a few minutes, but I think if you look at it carefully, you'll understand where the advantages come from.
Thank you very much, Dr. Kruppa. We really appreciate your time today. So just to remind everybody that any questions we didn't have time to get to will be sent to the speaker. And as with all the other talks in this event, the talk will now be available to view whenever you want for the next 30 days. And we will be back soon with the next talk.